All right, first thing we're going to do is we're going to review cell cycle or cell division. The first step of that is interphase. Remember, there's 3 steps to the cell cycle. The first one is interphase. What does interphase look like? Well, you know how good my drawing is, so let's use the tool. Okay, this is our cell. Now remember, during interphase, the cell is doing its normal life functions. If it's a skin cell, it's protecting. It's keeping homeostasis. If it's a muscle cell, it's moving. If it's whatever cell, it's doing its normal life functions, and it's also growing a little bit. The other thing, the main thing that it actually does is copies its DNA. Each one of these squiggly lines represents a DNA strand. Okay, like this one right here is a green DNA strand. It's made a copy of itself. Right here's the other one. They're identical copies, but they're in their DNA form. They're spread out. They're long and not very, not very put together. They're just a long line of DNA. Let's get another slide. Next part of the cell cycle is mitosis. Now, if you remember, mitosis is made up of 4 steps to accomplish mitosis. The first one is prophase. Draw my handy dandy cell again. So what happens during prophase? During prophase, these long strands of DNA condense. That means they get smaller. They're more tightly compacted. They're right next to each other. And all that information is really tightly wound up, and they join together to make a chromosome. A chromosome is 2 chromatids, okay? This right here is a chromatid, and this right here is a chromatid. Each chromosome has 2 chromatids, right there, right there, right there, right there, right there, right there. Okay. So each one of these DNA strands in the first stage of mitosis actually condenses itself down into these small X-type structures called chromosomes. These chromosomes are all held together by, they condense down into a X structure called a chromosome with 2 chromosomes being held together by a chromatid that holds them in the middle, right? Yes, Mr. Mansell. Okay, I forgot. Prophase is the first step of the oops, of the 4-step process. Okay. All right, so prophase is when the long strands of DNA condense down, make smaller into these chromosomal forms or chromosomes, which are 2 chromatids that are held together by a centromere. Okay? All right, we need another slide. The second step of mitosis is metaphase. Let me draw our beautiful cell since I can draw a perfect circle. Okay, during metaphase, that's when the chromosomes, DNA and chromosomal form, line up in the center of the cell. This makes it easier for the cell to separate them during the next step. Okay? All right, so metaphase, basically all it do is the chromosomes line up in the middle. We go to the next stage, or the next step of mitosis, which is anaphase. Get my perfect circle. Oops. Yep. All right, and during anaphase, what's going to happen is these chromosomes split, and they go to opposite sides. So actually those X's start looking like B's. They basically, the centromere that was holding them together breaks, and then they separate to other sides. These are now, instead of two complete DNA strands together, we have two complete separated DNA strands. The last step or phase batteries died. Can you believe that? Unbelievable. Timing is everything. Hold on to that thought. Why change the batteries? Do we have music? Da, 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 da. Da, 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 da. See if I can put them in the right way too. I didn't put them in the right way. You know, you have a 50-50 shot. And which way did I pick the wrong way? All right. Sorry about that. I'm going to start all over. So, the last step of mitosis is telophase. Telophase is basically the last step before cytokinesis, which is actually dividing. But in telophase, the strands of DNA, the chromosomes, they start to unwind. But they're unwinding in their own area. And also, nuclear membranes start forming. So the nucleus where the DNA stays, they start to um, form during telophase. And then the last phase of the cell cycle, the last stage is cytokinesis. And this is where cytokinesis, this is actually where the cells actually break apart. So instead of one extremely large cell, you have two. The cytoplasm of the parent cell divides into. And then you have two completely different daughter cells. Oops, not what I wanted. Two completely different daughter cells that have their own DNA. Wow, how did I miss the cell? They have their own DNA strand. They're identical to each other. And they're able to start the process new. And then these two cells would start going back into interphase. up an interphase that look exactly alike. Instead of one, you have two. Now these cells would start to grow, and as they grew, they would become more and more like a um, going into the interface step of cell division.